Our next presenter is DeirdreLogsdon. And Deirdre is a second-year PhD student in biomedical sciences. And with their degree, they hope to help mitigate the incidence of abnormal placentation while mentoring students in an academic setting. Their presentation title is Peering into the Window of Implantation.
When my daughter was younger, she used to see newborns wrapped in blankets, and she would think that those were called born babies because we would say that they were just born. And we can see a picture on the left-hand side in the background. That's a born baby. That's actually my son. And I got really lucky with these two. I didn't have any pregnancy complications, but it's estimated that 70% of pregnancies are lost before a live birth. And it's estimated that 30% of pregnancies are lost preclinically during a time when the mother doesn't even know that she's pregnant. This is a time when the embryo hasn't developed enough to produce the hormone necessary for a positive pregnancy test. Of those embryos that are able to attach, 3 to 7% of them will go on to develop placental pathologies like preeclampsia, contributing to intrauterine growth restriction and compromising the health of the mother and the fetus. What's so interesting about this window of implantation is it's so critical. That's when the embryo prepares for attachment, attaches into an endometrium, invades into a uterus in search of a maternal blood supply to nourish the fetus and the placenta for the duration of the pregnancy. But what I find intriguing about this is that even though it's so critical, so much is unknown about this window of implantation. It's unique to humans. We can draw inference from histological sectioning from decades ago and from animal models, but no animal model appropriately recapitulates the events that are happening during this window of implantation. Until recently, a group has developed an in vitro extended embryo culture system to see what's happening to embryos during the window of implantation without the uterus present. We've used this in vitro extended embryo culture system, and we've peered into the window of implantation and we've seen the unseen. So we can see embryos attached to the dish and proliferate and outgrow and start to differentiate the cells necessary for a healthy placenta. You can even see cells on the outside of the embryo begin to migrate away and look for a blood supply. And you can see a picture of that on the right-hand side. This is a picture that we've grown in our lab. This embryo, proliferative in the center, has a population of cells on the outer periphery that are going away searching for that blood supply. We've been able to take these embryos, digest them into single cells, and characterize their gene expression by cell type, and figure out what genes are controlling this process. How are they deciding to become one cell type or another? In particular, I'm interested in how these genes are being turned on and off in those cell types around the outer periphery. My hope is that by contributing to a broader understanding of normal placental development during this time, we'll be able to mitigate the incidence of placental pathologies like preeclampsia. We'll ultimately be able to help to reduce the number of early miscarriages and increase the number of healthy born babies. Thank you.